Okay, so let's start again with this idea of free energy. So this is, again, OpenStax, Chapter 6, Section 2. So Gibbs free energy is really a useful way of looking at how biological systems, how we can measure the energy that's being used and what is being actually available. So let's review these terms. So G is this idea of this free energy that's available, meaning that this is energy that we would expect then, depending on what the numbers are here, it would be usable energy, which would promote some kind of a reaction going forward or not. So that's what this delta G, remember delta means change in this usable free energy. Delta H is referring to the total energy in any system, total energy. Sometimes we look at this as enthalpy, for those of you who took that in chem. So this is the total energy. So we're looking at the change in the total energy of a system is going to, when we subtract from it the temperature and the change in the order of the molecules. In other words, this has to do with entropy or disorder. When we subtract this, the product of temperature times the delta S, or this entropy, that's what tells us how much energy is available for a chemical reaction to take place. So let's take a look at this. So what we're doing is calculating this. We're looking for this delta G. And we know that if that delta G value is less than 0, in other words, if this is going to be a negative value here, so when we've done the calculation, we end up with this being some negative number. That tells us that we have products that in the end, so here on this graph, you look down here, so if a chemical reaction happens, what we would expect then is that the products would have less available energy than the things that were the reactants. Now in biology, we frequently use the term substrates, and that's really referring to the reactants, the things that are going into a reaction. So when we have reactants, which basically have a higher energy potential, as you can see here on this graph, they have more energy than their products. And if we let this reaction go forward, and so to see what's going to happen, in other words, we're going to let this reaction happen, then what we know is that the delta G value is going to be less than 0. In other words, as the reactants react, then what we're going to get are some products which have a lower energy value, and we're going to have some energy that's going to be released. Now, in this, it's talking about spontaneity, spontaneous reactions. So think about that. That kind of makes sense to us sort of if we think about something happening in sort of quickly, we don't have to do anything. So a spontaneous reaction is generally one that will happen over time with or without any push. But you can see in this graph that even though we have reactants that have a high energy value, and they will eventually go forward, and we're going to get some energy released, we're going to get some low energy products, we would consider this a spontaneous reaction, and therefore energy is going to be released. This delta G value is going to be less than 0. So that's what we mean by spontaneous in biology. It's going to happen. It's going to happen because the reactants have higher energy value than the products. And so this is an important thing. Now, you can kind of see that there's a problem here, right? So we have to get over this hill. So we have to somehow get these reactants to release their products and that free energy. So let's see what happens. But in this case, we call these types of reactions are called exergonic. So exergonic reactions are considered spontaneous because we don't need to add any energy in to make them happen. but they don't necessarily happen quickly. So let's take the opposite situation. So let's say we have a chemical reaction in the body, and the reactants themselves have a lower free energy available to them than the products will at the end of the reaction. In order to get these reactants to form these products, we need to add energy in. So this is not a spontaneous reaction. Energy has to get added in. We're going to have a positive delta G value here. And that is called an endergonic reaction. Endergonic. So think like it goes within, ender, within. So endergonic reactions require energy in order for them to proceed, and they're not spontaneous. So when you look at these two, you can say, okay, are these reactions going to, so these two, here's our reactants, our substrates, these two here, these monosaccharides, and we're going to form this disaccharide. Is this reaction going to happen spontaneously, or is it going to require some energy? And then on the other side, we say, well, here is something that has, is the substrate. There's this disaccharide, and it's going to release to these monosaccharides at the end of the reaction. So is this one going to be spontaneous, or is it going to require energy? So think about it just in the terms that we just talked about. So when we have substrates or reactants which have a lower energy value than the products that they're going to form, this type of reaction is going to require some energy input in order to get a product that has more energy stored in it. And that's where that's stored is really here, right here in this bond. So in this case, this reaction would be endergonic. It means it's not spontaneous, it needs energy input. This one, however, we're going to release some energy by breaking these bonds. Here are the substrate, or the reactants, have higher energy value than the products, the individual products that they form. Therefore, this reaction here is a spontaneous one, and it's going to be considered exergonic. Now, you're logically saying, wait a minute, how does that all happen? Well, part of it has to happen, will happen over some period of time. Those exergonic reactions happen. However, in the human body, we don't always want to wait that long. So very often what we need is some form of something to help the process. And this hump that you see here in this diagram, this big hill that these reactants have to overcome, that's called the energy of activation, or activation energy. So this is the amount of energy that's kind of required for this reaction to go forward at all. So even though this reaction is showing something that is spontaneous, that is exergonic, which the delta G is going to be less than 0, that's how it's a measure of the energy that we're going to talk about here. But to go here, to get over this barrier, this is an activation energy barrier. It's really hard to get it going. What the body does is create enzymes, and those enzymes lower the activation energy barrier and allow that reaction to proceed. So this is why enzymes are super important. We need to get we actually kind of need to put the reactants in a little bit of an unstable position so that then they would be able to react. And that's a trick called a transition state. Let's get it to a point. It's kind of like you pushing a rock to the top of a hill. You're putting it in a position where it's a little bit less stable than it was sitting on the flat. And now we're going to be able to, with minimal effort, get that reaction to go forward. Now, heat energy is the main source for activation energy in a cell, And heat is going to help these reactants move toward this transition state. Now, it kind of makes sense too if you think about it, because heat energy, what does it do? When you heat molecules, they move more. So it's going to help them to potentially run into each other more often. Okay, and that's how a lot of chemical reactions happen in the body. It's the reactants basically are bumping into one another. So when we see something like this in this exergonic reaction, what we see here is the difference between a reaction that has a catalyst, an enzyme, so this is the catalyzed reaction, the green, versus the purple, which is that uncatalyzed reaction. So the idea, we'll talk more about catalysts and more about enzymes coming up. They're asking us to guess, but I'm just telling you, these are enzymes. These are what serve as catalysts to move a reaction more quickly forward. Now they ask you another question here. Is the rusting of iron happens slowly despite being a spontaneous exergonic reaction? So this is looking at the idea, or it's not really a question, it's just saying, well, here's an example of a reaction would happen slowly, but it'll still happen. And so that's one way to kind of look at that. Here's another example of an exergonic reaction. You can see here, we saw this one already, we see a hydrocarbon, some type of a fuel that's going to be used in this racing car. And so for this reaction to go forward, we provide that in the engine by creating a little spark that creates a little heat, and that helps that fuel, that hydrocarbon, to go over that barrier. And therefore this activation energy barrier is reduced, and then the products are formed, which would be just carbon dioxide, carbon monoxide, and water and things like that, which would be sort of the byproduct of combustion. Now, the laws of thermodynamics you spent a lot of time on, I think, in your physics class and probably even in your chemistry class. The only value in looking at this particularly is for some of the vocabulary that we've already used. So this idea of what is entropy, what is this concept of disorder, and what does it take to keep order in a system. And so I'm going to just go to this one slide here to tell you that one of the main source of energy for any of the reactions that require energy being added in is going to be that hydrolysis, or that breakdown using water of that molecule we've already talked about, ATP. So that's the main source of energy in most of these endergonic reactions that require energy being put in order to have that reaction go forward. This is ATP structure, so you can just kind of see it again. I'm going to quit right there because I think that's sufficient, and we will take up a little bit more. We'll get into more details on enzymes very shortly. So, hope that made sense.